Good morning, dear students. Today we are going to start our section with the topic. In the last video we just started different type of phospholipids. Phospholipids is a compound lipid, and in the last video we learned about lecithin. Lecithin, how that phospholipid become lecithin because of the presence of choline that we learned there in that video. Today we are going to learn about another phospholipid, that is phosphatidyl ethanolamine or cephalin. Or cephalin means it's another name. So from the name itself, it's very clear what is that additional group that might have present here. It is ethanolamine. Its structure is shown here. Look. Glycerol we can see to this one fatty acid, another fatty acid, our phosphoric acid, and we have learned that this much will make up the most simplest phospholipid, that is phosphatidic acid. And to this phosphatidic acid is coming that additional group here. In the case of cephalin, what is added, what get attached to that phosphatidic acid, that is ethanolamine. So because of that ethanolamine here, that makes that phospholipid a cephalin. Okay. So about that we are going to learn in this section. First, this cephalin differs from lecithin in that the nitrogen base is ethanolamine instead of choline. Okay. And where all we can find this cephalin? Where all? We can find in all biomembranes, and also it also possesses amphipathic properties. Amphipathic properties, we know that it has both positive and negative groups. So, or in other ways, polar and non-polar groups. And by that way, this cephalin also show amphipathic properties. In human physiology, it is especially found in the nervous tissue as white matter and also white matter. Then I don't know whether you know it or not. What is the difference between white matter and gray matter? White matter, as the name implies, it appears white because of the covering of myelin sheath around that nerves. And gray matter means it is devoid of that myelin sheath and it appears gray. So that's the one difference. And also this white matter mainly composed of the axon, nerve axon portion. We know that axon is mainly covered with the myelin sheath. And also as it is part of axon, it can conduct stimulus, means it can transmit, not conduct, transmit signal from one neuron to another. That is that area. Gray matter means it's mainly composed of cell bodies and it is receiving that stimulus. So that's the difference. I am just giving an additional information. I don't know whether it is additional or means already have information. Anyway, those who doesn't know, I'm just sharing this piece of information. Okay. So anyway, our cephalin is present mainly in the nervous tissue, especially where in the white matter, and also spinal cord, nerves, neural tissues, and here all we can see this cephalin. 45% of the phospholipid present in all these areas are of cephalin. Then what's the biological significance? Biological significance of the cephalin. Phosphatidyl ethanolamine or cephalin, it play an important role in membrane fusion. Membrane fusion means what? Membrane fusion means we know that, I don't know whether you know it or not. Anyway, you inside the cell, because means for secreting so many substances to out. Some vesicles are coming towards the plasma membrane and it is fusing. So and it is one instance only. In likewise, in so many instances, this membrane fusion is required. So they all what plays important role? Cephalin is required. So in those all areas of the membrane where the membrane fusion required to take place, there all cephalin is abundant. And also it is abundant and it helps in the disassembly of the contractile ring. Contractile ring is a ring that is forming and it causes a cell to divide into two daughter cells by forming a ring here. And that ring, you might have learned it somewhere else, some other chapter. It is formed of actin and myosin proteins. And as this ring form, it constrict, and it meet each other and will divide that cell to two, this single cell to two daughter cell. So as these two constriction meet, then the contractile ring need to disassemble. So that time also this cephalin is very important here in this type of process. In here in this area means that area of membrane also rich with this cephalin. It is helpful. It is very important in the disassembly of the contractile ring during the cytokinesis. Cytokinesis in the cell division. Cytokinesis means division of the cytoplasm. And the cytokinesis, we know that it happens at the end of the cell division. And because of that, the cell is dividing. Anyway, leave that. And that's not our topic. Anyway, the disassembly of the contractile ring, in that also the cephalins are very important. Then again, this cephalin regulates membrane curvature. Membrane curvature is the same process. I already explained you at the time explaining about membrane fusion. So many curvature, so many shape change need to have that cell during different physiological process that is happening inside the cell. In all those time, those areas where need to undergo some curving or curvatures to facilitate some important process, in those areas are all rich with this cephalins. Okay, or phosphatidylethanol amine. Membrane curvature refers to physical bending of membranes to accommodate various cell morphological changes. That's all I need to understand. So, cephalin, its importance we have seen. Next, phosphatidylinositol. Look to its name. Here, what is there? What the name implies what could be that special group? Inositol. So, our glycerol is here, two fatty acid, phosphoric acid, and this much will form phosphatidic acid. And to this, what is attached? Inositol. So that is another phospholipid. It is a phosphatidic acid is esterified to inositol. Okay. Next is another one more variant form is present, that is phosphatidyl inositol bisphosphate. Listen to that for bis means two phosphate group. Here this is the structure. Two phosphate group are attached to fifth and fourth carbon of this inositol ring. So that is why phosphatidyl inositol bisphos, because it is that phosphoric acid are attached to nearby carbon. So that's why called bis instead of di. So phosphatidyl inositol bisphosphate, it is also present in biomembranes and also in one more place you might have learned this name. Where it acts as a second messenger, right? Second messenger in hormone mediated cell signaling pathway. So there also it has some role. It acts as a second messenger to bring about some process inside the cell. So that is the significance of phosphatidylinositol. I hope that its structure and its significance is clear to you. Next, we are going to another type of phospholipid. What is it? Plasmalogens. Plasmalogens means what? So that's also very interesting. Here, we need to understand that here one fatty acid is there. The difference is that here an alcohol, unsaturated alcohol, is attached to the first carbon, and that attachment is through an ether linkage. So then at this context, we need to understand between difference between ether and ester linkage. Ether linkage, for making an ether linkage requires only one oxygen and two carbon. Look, ester linkage for making such a linkage, two oxygen and two carbon is required. So that is the difference. Plasmalogens, here what? And also this alcohol is unsaturated. Look. Alpha and beta carbon is unsaturated. So unsaturated alcohol is attached to the first carbon of glycerol through what linkage? Ether linkage. And along with that, the second carbon is through ester linkage connected to another fatty acid. And phosphoric acid is here, and along with that, this X represent, it can be either choline or ethanolamine. Any one of these things can come and join with us with this bond. So see, these are phospholipids which have an aliphatic. Aliphatic means straight chain, long chain, alpha beta unsaturated alcohol in ether linkage with the first hydroxyl group of the glycerol. So the alcohols have about what would be its length? Maybe 12 carbon or 18 carbon will be the chain length of that alcohol. And the second carbon is esterified to fatty acid. And that phosphoric acid can be either bonded or connected to choline or ethanolamine. So that either it is connected to a choline or ethanol in both cases, both type will be a plasmalogen itself. Okay. Then where we can find plasmalogens? It also important compound and constituent of biomembranes, where brain and the muscle. And also nearly 30 to 40 percentage of human heart tissue, it is composed of this plasmalogen. Plasmalogen, which type of plasmalogen? Choline attached plasmalogen. So as it is attached with the choline, in an expanded way, we can call it as this plasmalogen as choline glycerophospholipids. We know that phospholipid and glycerol will be there, and here instead of ethanolamine, choline is attached. So that type of plasmalogen is present in the human heart tissue up to what percentage? 30 to 40 percentage. Next, we are going to see another type of phospholipid, phosphatidylglycerol. What is what is its peculiarity? The name itself is clear that an additional glycerol is attaching here along with our phosphatidic acid. Look here, this is glycerol, the first fatty acid, second fatty acid, phosphoric acid. This up to here, this will form phosphatidic acid, and to this again attach one more glycerol. So that will form the phosphatidylglycerol. And it is formed by the esterification of phosphatidic acid to 1-glycerol. Phosphatidic acid to 1-glycerol. Then such a phosphatidylglycerol example is cardiolipin. What is its peculiarity of cardiolipin? Here, 2-phosphatidic acid, it is linked with the 1-glycerol. So that will form the cardiolipin. 2-phosphatidic acid. linked with the glycerol. That forms the cardiolipid. When two molecules of phosphatidic acid linked with a molecule of glycerol, then diphosphatidylglycerol or cardiolipin is forming. It is a major phospholipid of where mitochondrial membrane. It is present in the mitochondrial membrane. So it's very important. We know that mitochondria is the ultimate organelle to release energy and it should be perfect. Okay, and that's the significance of cardiolipid. Commercially, we can extract from myocardium and decreased level of cardiolipin. It will lead to mitochondrial dysfunction. Mitochondria means doesn't function properly. It may result in heart failure. We know that cardiac muscles also need enormous amount of energy to continuously beat, and the ultimate energy releaser is mitochondria. And as the cardiolipin level is low, it cannot function. It will not provide enough energy, and heart may failure. And other means conditions, associated conditions. So many associated conditions also there. Hypothyroidism and so many myopathies are there. So that's the significance of cardiolipin. Okay, so we finish different types of phospholipids. Then this is another type of compound lipid, sphingolipids. What is the difference? Here we know that basically phospholipids, the alcohol, glycerol is that ultimate part, is that alcohol part. But here, instead of glycerol, sphingosine is present instead of glycerol. So we can see this is sphingosine. So it is a lipid, compound lipid, and its backbone. Backbone is formed of aliphatic amino acid. Its name is sphingosine. Instead of glycerol, what type of alcohol is present in sphingosine? Aliphatic amino alcohol. This picture is shown here. Aliphatic, straight chain, then amino group is there, and also alcohol. Amino alcohol, aliphatic amino alcohol. Example is that aliphatic amino alcohol is sphingosine. So sphingosine containing lipid, that forms sphingolipids. Ceramide is a type of sphingolipids. When ceramide is forming, when the sphingosine combined with the fatty acid through amide bond, sphingosine is attached to fatty acid through amide bond, that will form a ceramide. So this is the amide bond, C double bond or NH. So through amide bond, when sphingosine is attached with a fatty acid, then it will become ceramide. Okay, and there are so many lipids that contain the sphingosine in it. They are phosphosphingosides. The name implies that contains phosphoric acid. Then glycosphingolipids, the name indicates that carbohydrate will be there. Sulfatates means sulfate group will be there. Okay, so let's see one by one. First, the phosphosphingosides. They contain phosphoric acid here, then sphingosine, then one fatty acid. So we have just learned that sphingosine with the fatty acid that forms ceramide. So along with the ceramide, one phosphoric acid group is there and a chloride group. So that forms that phosphosphingoside. And a common type of phosphosphingoside is sphingomyelin. From the name itself, it is clear that it is present in myelin sheath. Okay, it is a common phosphosphingoside present where in biomembranes, especially nervous system, and more specifically seen associated with the myelin sheath. And different type of sphingomyelin is possible to occur. How different type may form depends upon the difference in the fatty acid group attached to that sphingosine. As this fatty acid group changes, different type of sphingomyelin is forming. Commonly, which are the different types of fatty acid binding with or attaching with this sphingosine? They are lignoceric, the fatty acid, 24 carbon long, nervonic, 24 carbon with one double bond, and servonic. These are all different fatty acids that are connecting with the sphingosine group to create to form different type of sphingomyelin. Then what is the significance of this sphingomyelin? It is amphipathic. This one also amphipathic in nature, and it also an emulsifier mixer. It can mix two substances thoroughly together, and also it is a detergent. And one more importance, I have already told you in the former video about lecithin that how doctors understand the healthy development status of embryo by checking the lecithin sphingomyelin ratio of the amniotic fluid. And at that time, I told that it should be 2. If it comes down, it indicates that the lungs is not working properly. So anyway, the relative proportion of lecithin and sphingomyelin is important in biological fluids like bile, amniotic fluid, etc. Next, we are going to see another sphingosine, that is glycosphingolipids. It is always called glycolipids, means carbohydrate containing lipids. And also it doesn't have phosphoric acid group, non-phosphorylated lipid, this one. So it is a sphingosine, and also it is a glucose containing one, and it doesn't have phosphate. So here the lipid doesn't contain phosphoric acid. That is what difference. Instead, it contains carbohydrate and ceramide. Ceramide, we know that it is a combination of sphingosine plus fatty acid through amide bond. Carbohydrate and ceramide. So, ceramide plus glucose, monosaccharide. This will form glucose cerebroside. Then another monosaccharide, ceramide plus galactocerebroside. Then globoside means what? Globoside means along with the ceramide, instead of monosaccharide, oligosaccharides are there. Means it contains more than two. It contains 2 or more monosaccharide units along with the ceramide. They contain 2 or more hexoses or hexosamines attached to ceramic molecule. So here ceramic plus two hexoses, galactose and glucose. That forms lactosyl ceramide. And this lactosyl ceramide is important component of erythrocyte. Erythrocyte membrane is mainly composed of this glycolipid, lactosyl ceramide. Then ganglioside. Ganglioside means what? Ganglioside means it is formed when ceramide will be there, oligosaccharide will be there, and it is combining with the NANA. NANA means N-acetyl nuraminic acid or sialic acid. So, only one difference. When to this globoside, when NANA is attached, then it will become ganglioside. Clear? So, ganglioside means ceramide will be there, oligosaccharide will be there, and one molecule of NANA, N-acetyl nuraminic acid or sialic acid. So, that's what we are seeing here. Ceramide, glucose, then galactose 2, hexose group, then NANA. It is designated as, it is in short form, it is designated as GM3 or ganglioside M3. Ganglioside M3. What is its significance? Its main significance, it gives stability for paranodal junction. Then what is paranodal junction? Paranodal junction means This suppose this is the axon, and around the axon, myelin sheath is there. Myelin sheath is formed of Schwann cell, and the Schwann cell, it is tightly attached with the axon by bending, and that point, that area is called paranodal junction. So it gives stability to that paranodal junction, and also give stability to the ion channel clusters and that present in the myelinated nerve fibers. So that is very important. It's very important. Our nerve should be intact and stable. Only then we can receive, we can do anything. So that much. Then we are moving to the last type of sphingosine. It's sulfolipids or sulfatate. As the name indicates, it contains that sulfate group. Sulfate. Here we can see that sulfate group is there. To fatty acid sphingosine base is there. So that main thing is that it contains a sulfate group. Sulfate, it is synthesized. Its synthesis starts in the endoplasmic reticulum, and we have learned that the smooth endoplasmic reticulum is concerned with the synthesis of lipids. So this is one lipid. So synthesis starts in endoplasmic reticulum and ends in Golgi. And on reaching Golgi, it will be in the form of ceramide, and it is then converted to galactocerebroside. And after it become galactocerebroside, it is sulfated to form this sulfatate. This is the way how it is forming. So what is this sulfatides or sulfolipids? They are formed when sulfate groups are attached to ceramide oligosaccharides. Ceramide we know what are oligosaccharides. Ceramide plus oligosaccharides will form globosides. Okay. Then along with that, if sulfate group is there, then it will form sulfolipids. What is the significance? It is the important component of, again, nervous tissue. So most of the lipids are very important with the nervous tissue. And when its level become high, and it is very essential to degrade to reduce its level. If the degradation doesn't happen, it will accumulate in the central nervous system, and that condition is called the lipid storage disease. Okay, so these are the points associated with the sulfolipids. Next, we need to know what is lipidomics. Lipido. First, we need to understand what is lipido. Lipido means it is a total lipid that present, total lipid that present in the organism is called lipido. And the study of the structure and function of all members of the lipidome. Need to study all lipids, all members of the lipidome, both during in healthy situation and also in diseased condition. That is lipidomics. So lipidomics is simply the study of a study about all the lipids that present inside one organism. When that organism in healthy and also in disease condition. And one more thing I need to make you clear up to where we have finished so far. We covered simple lipids. It is ester of fatty acid and alcohol. And now we are talking about compound lipids. Compound lipids means composed of fatty acid, alcohol plus some additional group. We finish phospholipids, glycolipids. Then need to learn about lipoproteins that we'll discuss in the coming sections. Okay, with this I'm going to finish this section. Thank you. I'm an icon, man. I'm a girl about a 10. I should fuck her in a Benz.